Hello, this is Dr. Bob Remedi, and I am here to talk to you about the last lab of the semester for Biology 244, which is the Special Senses Lab. So we're going to be looking at the eye and vision and the ear and hearing throughout this lab. So let's get started. We're going to start by looking at the eye. Here is a picture that's showing some of the accessory organs that are associated with the eye. So you can see over here we've got the eyebrows, those series of hairs that are used to help protect the eye from sweat trickling down into your eye from your forehead. We have both eyelids and eyelashes. There's the lower lid and the upper lid here. Eyelashes do some of the same job as the eyebrows. They help keep out dust and dirt and insects and stuff like that, hopefully trapping them and preventing them from getting into the eye. The eyelids are also going to provide a physical barrier for the eye. In addition, a structure that we can't see here, one we'll see on the model, is the lacrimal gland. When the lacrimal gland produces tears, those tears are going to be moving from the lateral aspect of the eye to the medial aspect of the eye, helping to wash away dust and dirt and such. And the eyelids, when you blink your eyes, are also going to help spread the tears of those lacrimal fluid. And then you can see really nicely we've got a number of what are called extrinsic eye muscles. And the extrinsic eye muscles are responsible for moving the eye so that you can track things up, down, left, right, and all around too. Okay, looking at a section through the eye. This is actually a cross section through the eye where you've separated. You can see the inferior aspect of the eye, the superior aspect has been removed. So it's like you're looking down into the eye here. And we can see that there are three main layers of the eye. And I've split those main layers. If you look into the corner here, the most superficial is called the fibrous tunic. Deep to that, we've got the vascular tunic, and then we've got the retina. And I've color-coded some of the structures here because each of these layers is going to be split up into some different parts. And then I've got some things in yellow here that we'll talk about in greater depth that can be a little challenging sometimes. So the most superficial layer of the eye is the fibrous tunic. Fibrous tunic is split up into two main parts. The more anterior portion of the fibrous tunic is this clear, colorless layer called the cornea. The cornea is going to allow light to travel into the eye, but helping to keep out, again, the dust and dirt and all that stuff we don't want getting into the eye itself. Okay, the posterior, probably 2/3 or so of the fibrous tunic is going to be called the sclera. And the sclera is sometimes referred to as the white of the eye. Now, if you're a student of American history, perhaps you remember Colonel William Prescott, who during the Revolutionary War told his troopers, "Don't fire until you see the whites of their eyes." That was because the smoothbore muskets that they used at the time were not particularly accurate. So by the time the enemy is close enough that you can see the whites of the eye, your musket was probably accurate enough to be able to hit somebody. So that is our fibrous tunic, the more superficial layer. Deep to the fibrous tunic, we've got the vascular tunic. And there are a number of different parts of the vascular tunic. We're going to start posterior here and talk about this layer called the choroid. Actually, let's back up a little bit. So the term vascular, hopefully you know by this point, means vessels. So the vascular tunic has lots of blood vessels in it. The fibrous tunic does not have any vessels in it. You certainly wouldn't want any blood vessels traveling through the cornea here. That would block some of the light going into the eye and would cast a shadow onto the retina, which would make it hard to see things. But the vascular tunic has a lot of blood vessels in it, and the most posterior part of it is called the choroid. Choroid is this brownish or blackish layer, and the pigments in there are to absorb extra light. So light doesn't just bounce around on the inside of the eye and throw off your sight picture. The more anterior portion of the vascular tunic is called the ciliary body. And the ciliary body's got some other parts and pieces that we'll talk about a little bit more in the next slide when we look at the more anterior part of the eye a little bit more close up. Okay, the deepest part of the eye is called the retina. And the retina, you can see here, is this layer that's going to be containing the rods and the cones, which are the cells that are responsible for detecting light. We call them the photoreceptors. And the axons of neurons that are going to synapse with these rods and cones are then going to be leaving the eye through this area here, which is the optic nerve. So there are two types of fluids that we're going to run into. In the anterior cavity, or excuse me, the anterior segment, was sometimes called the anterior cavity, is going to be filled with this thin, runny fluid called aqueous humor. Okay? The posterior segment is going to be filled with this thick, viscous fluid called vitreous humor. And we're going to talk about the significance of both of those shortly. But do know that that thick, vitreous humor here is going to help hold the retina up against the other layers of the eye. If you've ever heard of somebody having a detached retina, literally what happens is the retina pulls away from the vascular tunic here, and that can result in blindness if you're not careful. So vitreous humor is the thick stuff on the posterior segment. Aqueous humor is going to be found on the anterior segment here. Zooming in and looking at the anterior segment more closely. We're gonna make things a little bit more difficult because the anterior segment is actually split up into two parts. But let's talk about the anterior segment in general first. So the anterior segment is going to go from the cornea here to the lens. And as I mentioned before, that this is going to be filled with that runny aqueous humor. Posterior to this is going to be the posterior segment, and this is the area that's going to be filled with vitreous humor. So we're going to split up the anterior segment into, ready for this? The anterior chamber of the anterior segment and the posterior chamber of the anterior segment. I know, don't blame me, I didn't come up with this, but it's what we have to work with. So the anterior chamber of the anterior segment is going to be the area here from the cornea to the iris. Okay. Posterior to the iris. So we've got the iris here and we've got the lens here. This little region right here is going to be between the iris and the lens is going to be the posterior chamber of the anterior segment. Okay? And the aqueous humor is going to be made in the ciliary body in here, particularly an area called the ciliary capillaries, which you can't really see here, but they would be here. So fluid's going to leak out of these ciliary capillaries, and they're going to go into the posterior chamber of the anterior segment, and it's going to wash over the anterior part of the lens, come through the pupil, this is the hole in the iris here, come through the pupil, and it's going to wash down through the anterior chamber of the anterior segment, and it's going to drain out these little holes. You can see one here. You can see the other one here. These are called the scleral venous sinus, or the old-fashioned term for that is the canal of Schlemm. And that's where you're going to drain out extra aqueous humor. If something were to happen and the scleral venous sinus were to decrease in size or be plugged up completely, aqueous humor would build up here, and that would result in a lot of pressure, kind of like filling up a water balloon. That pressure would push on the lens, which would push on the vitreous humor back here, and that would cause a dangerous increase in pressure in the eye that we know of as glaucoma. This is a picture of what your retina looks like. So when you go to the optometrist or the ophthalmologist and they look into your eye to get a picture of what your retina looks like, this is what they see. Now, all the little tree branch-like things that you're seeing over here, these are capillaries in your retina. You got to make sure your retina is well vascularized. And we see a number of things. So off to the left side of the picture here, you see this area called the optic disc. This is the part of the retina where the ganglion cells that are attached to bipolar neurons, and the bipolar neurons are attached to the rods and cones. This is where they leave the eye. So this area right here does not actually have any rods and cones in it. So a little later on in the lab, you'll do a little exercise to see your blind spot. So because the optic disc doesn't have any rods or cones in here, this is responsible for your blind spot. The other thing that you see right here, it just so happens that it's in the center of your field of view, and I put a circle around it. This is called the macula lutea, and it's hard to see in this picture, but it's kind of a yellowish colored structure. That's what lutea means, is yellow. And in the very center of the macula lutea is a depression called the fovea centralis. And the fovea centralis is a little cup-shaped depression in which we have the highest concentration of rods and of cones in your retina. And by using those extrinsic eye muscles to point the fovea centralis at the light that's coming directly into your eye, something that you want to see with high resolution, that high density of cones is going to allow you to see with a lot of sharpness. Okay, so the fovea centralis is that little pocket full of cones that gives you your highest visual acuity, the greatest sharpness. This is an interesting picture because it's talking about some of those neurons that I was mentioning before. So here you can see the cone cells. These are responsible for detecting color vision, and there are different cones for the different primary colors. And then these cells are the rod cells, and they're not so much for detecting color, but they're for detecting light and lower light levels, so for black and white vision. And I've got this eye picture here kind of oriented in the direction in which you would be seeing it. So light would actually be entering into the eye through the cornea, through the pupil, through the lens, and the lens would be focusing the light onto the retina here. But isn't it interesting that the light has to travel past the ganglion cells, past these bipolar neurons, to get to the rods and the cones, which are really facing the posterior aspect of the eye? You gotta wonder if this is efficient enough to be able to see, but since most of us do have vision, this must work okay. But the rods and the cones, as you see here, they're going to synapse with the bipolar neuron. The bipolar neurons are going to synapse with these ganglion cells. The ganglion cells are going to come together at the optic disc and actually leave the eye through the optic nerve and then make its way to your brain, where you will interpret these action potentials as sight. One of the things we ask you to do in the lab is to use Ishihara's color blindness test. Well, if you are at home and you don't have access to a Ishihara test book, I'm putting up some typical pictures that you would use for this. These are made of different colored dots to see if you are able to differentiate between the different colors. So looking at this one right here, many of the dots are kind of green and bluish, but they've got some kind of peachy orangey. And if you can see, this one says 40 and 5. So to determine if somebody has color blindness, you would use a chart very similar to this.
So here you see two different models of the eye. I've chosen both of these models because each one shows slightly different things. So before we get to talking about the parts of the eye itself, there are a few accessory organs we need to look at. The first of which is this part right here, which is a lacrimal gland. Lacrimal glands are going to be secreting tears, and you find them on the lateral aspect of the eye, and when they're releasing tears, the tears are going to be moving medially and inferiorly, kind of washing any dust, dirt, and debris away from this clear layer here called the cornea. We'll be talking about that in a moment. And then eventually making its way to the lacrimal gland, which would then be draining it away. You can also see on this model really, really clearly that we've got extrinsic eye muscles. These are the muscles that are going to attach to the sclera, or the white of the eye, and by contracting them, it allows the eye to move laterally, medially, up and down, etc. All right, so the eye itself is made up of 3 layers. The most superficial layer is called the fibrous tunic. Fibrous tunic is made of connective tissue and it is avascular. Our fibrous tunic, we've got the sclera or the white of the eye, and we've got the cornea, which is the clear colorless part. And both of those make up the fibrous tunic. On this model over here, you can see that I've got part of the cornea intact here, part of it has been removed, so you can see some of the deeper stuff, part of the vascular tunic or the iris. Okay, so the layer that is deep to the fibrous tunic then is going to be called the vascular tunic. The vascular tunic is going to include, here we see, the choroid, which you can't really see here because the deepest layer of all, which is called the retina, is intact. But if we would peel back the retina, there would be this dark brown layer here called the choroid. And it's dark brown or sometimes black because that's going to prevent any light from striking the inside and then bouncing around and hitting the retina in a weird way. So the other part of the vascular tunic is called the ciliary body. And it's within the ciliary body that we're going to find muscles called ciliary muscles, in addition to suspensory ligaments that are going to attach to the lens. And by contracting the by contracting the ciliary muscles, it pulls on the suspensory ligaments and changes the shape of the lens. And by changing the shape of the lens, it's going to allow you to focus light on the retina differently for seeing things that are close up or for seeing things that are far away. In addition, we see here the iris is going to be part of the vascular tunic. And the iris is the colored part of the eye, and it's the hole in the iris which is called the pupil. And we did some pupillary testing in the reflexes lab, which hopefully you remember. Okay, in this model here, you see the lens. Now in real life, the lens would be kind of squishy, right? So the suspensory ligaments and the ciliary muscles that would attach to it, by contracting those ciliary muscles, it would change the shape of the lens and as a result change where you're focusing. When we do dissections on cow eyes or sheep eyes, the lenses have been soaking in a preservative, so they're often hard, and certainly in this model, this is really hard. Okay. Now the liquid that would be circulating in the more anterior part of the eye is the aqueous humor. We can see this part of the eye right here. It's plastic and it's shaped like a semicircle here. Well, this is to represent the very thick, viscous part of the eye called the vitreous humor. And the vitreous humor is going to be found in the posterior segment of the eye. Okay. The deepest layer of the eye is going to be the retina. And you can see the retina very clearly in here. It's going to be this off-white, or in the case of this model, it's kind of an orangey color. And the retina is going to contain the rods and the cones, which are the light-sensitive neurons for detecting light. Now, if you see, let me see if I can angle this the right way. There is, in the posterior part of the eye, this kind of yellowish colored area here, which is called the macula lutea. And in life, it would be kind of a yellowish color. There is a depression in the macula lutea called the fovea centralis. And the fovea centralis is this depression in which you would find the highest concentration of cones. Now, the cones are the cells that are used for detecting color vision. And by having a dense concentration of them, that would give us the greatest acuity or the greatest sharpness. So when we're looking at something, and let's say something we want to read in a book, okay, we would be using our extrinsic eye muscles to be turning our eye so that light goes through the cornea, goes through the pupil, goes through the lens, and the lens would focus what we wanted to see with the highest acuity on the fovea centralis here, and that would allow us to read whatever it is that we were reading and see it clearly. Okay. What we can't see real well in this model, we can see quite nicely in this model right here. So in that deepest layer, the retina, all the neurons that are coming out from the retina, the rods and the cones are going to synapse with bipolar neurons. And the bipolar neurons are going to synapse with ganglion cells. And those ganglion cells are all going to come out the eye in an area here called the optic nerve. So here we've taken the smaller eye apart, and you can see really clearly here's the retina. Okay, but the thing I really want to point out in this one is where all the rods and the cones that are leaving the retina are going to synapse with bipolar neurons. Those bipolar neurons are going to synapse with ganglion cells, and all of those ganglion cells are going to leave the eye as the optic nerve. Where the optic nerve leaves the eye, there's actually no rods and cones. So we call this area here the optic disc, and the optic disc actually, because there's no rods and cones there, represents an area in our retina where we do not see. And if you follow along in the lab book, there's a little test to show you that in fact you have a blind spot, which fortunately, because we have two eyes, our brain doesn't allow us to interpret most of the time.
Now that we finished up the eye, we're going to start talking about the anatomy of the ear. So here's a diagram of what the typical ear looks like. Ear is split up into three main parts. We've got the external ear, the middle ear, and the inner ear. As we'll see on the models in a few minutes, the external ear is made up of the auricle, also called the pinna. This acts kind of like a funnel to help collect sound and to direct it into the tube here called the external auditory meatus. Sound waves are then going to strike the tympanic membrane that you see here, and the tympanic membrane is going to vibrate. Vibrating the tympanic membrane is going to cause those vibrations to travel along the three bones, the malleus, the incus, and the stapes. So the malleus looks kind of like a hammer, and in fact malleus means hammer in Latin. The vibrations from the tympanic membrane cause the malleus to vibrate, which causes the incus or anvil to vibrate. And those vibrations are then brought to the stapes. And stapes is the Latin word for stirrup, which equestrian people know is where you put your foot when you're riding a horse. And the vibrations from the stapes then go into this little depression in the temporal bone called the oval window. And that's going to be bringing the vibrations to the inner ear. Okay, the middle ear is the area between the tympanic membrane and the temporal bone here. Now perhaps you've had a middle ear infection, otitis media, or you know some little ones that have had chronic ear infections. That happens when you've got some pathogens in here. And when you've got bacteria or virus growing there, the body's natural response is inflammation. Well, inflammation, normally in the middle ear here, the pus and extra fluid would move down sometimes it's called the eustachian tube. More commonly, we call it the pharyngotympanic or the auditory tube. However, especially in younger people, the pharyngotympanic tube kind of closes off. So this pus is allowed to build up, and when that happens, it pushes on the tympanic membrane here. That is very painful because there's a lot of nerve endings in the tympanic membrane. So the surgery for this is to put a little grommet in here, a little tube that will allow any pus to travel through the tympanic membrane and out until, oftentimes as children get older, the pharyngotympanic tube opens up and then you can drain pus this way. This is also very important in rapid pressure changes. So if you've ever flown in an airplane and you do the Valsalva maneuver, where you blow on your ears, what you're doing is you're forcing air into the pharyngotympanic tube to hopefully equalize the pressure between the middle ear and the external ear. Okay, let's see another picture and look a little bit greater depth in the inner ear. So this is kind of a neat picture. It shows you the tympanic membrane here and the malleus, the incus, and the stapes. Now they've completely removed the temporal bone, so we can see the inner ear in much greater detail. And there's three important parts to the inner ear. We've got the vestibule in this area right here. We've got the cochlea, and we've got the semicircular canals. The cochlea here, cochlea is taken from the Latin word for shell, snail shell. And this is this great big long tube that has been rolled up into this snail shell structure. And it's within this structure that we're going to find the sensory neurons that are responsible for detecting sound. And we've got the semicircular canals and the vestibule, which are going to have sensory neurons to help us with balance. Okay, whether the sensory neurons for balance, the vestibular sense, or we've got the cochlea here, the action potentials for sound. These are then going to travel out the vestibulocochlear nerve, maybe you remember from the cranial nerves here, along this nerve right here. Now, something I'd like to point out, you can't see the oval window here very well because part of the vestibulocochlear nerve is covering it up. But you can see this little structure here called the round window. And as sound waves are going to go into the cochlea, and I'll show you another picture of that in a few moments, any wavelengths of sound that are not going to be used to generate action potentials actually travel out the cochlea and back into the middle ear through this narrow region called the round window. So this is a different picture. It's showing the middle ear, and it's color-coded a little differently to show some structures that we talked just a tiny little bit about in the lab book. We mentioned the term bony labyrinth and membranous labyrinth. So the more superficial portion of the inner ear here is going to be what we call the bony labyrinth, and it's going to be filled with a fluid very similar to interstitial fluid called perilymph. And then the deeper aspect of these organs is going to be what we call the membranous labyrinth, and that's going to be filled up with a different liquid called endolymph. But the important thing to remember is that the inner ear is going to be filled with fluid. Now, this is a really cool picture because the cochlea has been unwound into this straight tube, and it's broken down actually into 3 different tubes. So when sound waves are going to come in from the stapes to the oval window, these vibrations are going to be traveling into the cochlea. And this long tube is going to contain sensory neurons that are sensitive to vibrations or sound. Where we have sensory neurons that are sensitive to a particular wavelength of sound, they're going to be generating action potentials in this inner portion of the cochlea called the cochlear duct. And then any vibrations, any sound that is not going to be detected in the cochlea are then going to be traveling out through the round window so that they're not going to interfere with new sound waves coming in. And that's the diagram of the ear. So let's talk about, and let's show you some examples of the parts on an ear model.
So here you can see we have the picture of the ear model, similar to the one that we would be using in class. So the first structure that you see right here is the ear pinna, or the auricle. This acts kind of like a funnel to collect sounds and then to direct it into the external auditory meatus, which should sound awfully familiar since that was the name of the hole in the temporal bone that's going to allow sound waves to get to the middle ear. So at this point, the external ear represents the auricle and the external auditory meatus. This next structure that we see right here is the tympanic membrane, and this is what vibrates when sound hits it. Tympanic membrane is going to be the boundary between the external ear and the middle ear. If we pull the tympanic membrane out, here you can actually see through it one of the ear ossicles, the bones that are going to connect to it. And that first one is going to be the malleus, or the hammer, because it looks kind of like a hammer. The second is the incus, or the anvil. And unfortunately, the third bone, the stapes, broke off on this model, and I wasn't aware of it. So there would be a third bone connected to the incus called the stapes, which looks like a stirrup. And the stapes is actually going to vibrate against the temporal bone, and that's going to be bringing sound waves into the inner ear. Okay? I'm going to keep the tympanic membrane out of the side, off to the side here, so that you can then see the inner ear. And the parts of the inner ear that we're going to talk about include the cochlea, the vestibule, and the semicircular canals. So the vestibule is the area, it's kind of like the common area or the vestibule in a building. And we see the vestibule going off and connecting with the cochlea. Cochlea is taken from the Latin word for snail, and it's this snail shell shaped structure. You can see it a little bit better here. The snail shell shaped structure that, if you were to uncoil it and pull it out, would be where we have the mechanoreceptors that are responsible for detecting sound. From the vestibule, you see right here, we have these three kind of semicircular looking things. Those are called, oddly enough, the semicircular canals. And inside the semicircular canals, we're going to have mechanoreceptors that are going to be detecting movement. The fluid inside of the semicircular canals, when it sloshes around, it's going to bend hair cells, which is going to generate action potentials, and then would be going out the vestibular branch of the vestibulocochlear nerve to bring the sense of balance to the brain. The mechanoreceptors in the cochlea would be attaching to the cochlear branch of the vestibulocochlear nerve, and they would be bringing information about sound to the ear. And that's our ear model.